the data that you have. So a typical form factor would be an ellipsoid, and one can, by changing the axis of the ellipsoid and computing the theoretical scattering data from this ellipsoid, one could try to approximate the experimental data. However, that would be a very rough approximation, but it is used in, so a more complicated version of this is used when you have, for example, lipids and proteins together, and you need to model that. That is fairly sophisticated. However, bodies is like a very low resolution overall approximation of the shape. And let us look at something more interesting. And so there is an approach that is called dummy atom models. So what is a dummy atom model? So a dummy atom is a sphere, like a bead, like a small, very small sphere. And out of these beads, or dummy atoms, you may construct a shape that would be the low resolution shape of the particle of interest, for example, of a protein. So how is this done? So we take a large sphere, usually of the size of Dmax, that we estimated from the P of R step, and we fill it with densely packed beads. And each bead can be either a solvent, or like empty space, or a particle. So, and by shaking these beads and like changing each bead from 0 to 1, where like 0 is solvent, or like nothing, and 1 is the particle, we can come up with a shape that, if you calculate the theoretical scattering from the shape, it would fit the experimental data. So there is a quick way of computing the scattering called an approach called spherical harmonics, which will give you an approximation of the scattering which is good enough for our purposes. So this is a computational method that permits us to quickly compute the theoretical scattering pattern from this bead model. So, and this is a Monte Carlo type search, so which means that it is like you randomize your model and you try to change it in a way that it would change the scattering pattern towards minimizing the discrepancy between experimental data and theoretical scattering. And we use a procedure called simulated annealing, which permits us to avoid the local minima. So we kind of always, using this method, we always fit the experimental data. However, so we have a program called Damin, and this program is an implementation of this algorithm. So however, we can come up with various shapes that would fit the experimental data equally well. For example, these three shapes all produce the same scattering pattern that is a good fit to the initial experimental data. However, one of them is disconnected. So that means that it is not possible to have a disconnected protein, right? It should be all interconnected body. And the other one is too loose. So it has some fluffy tails sticking out. And we know that provided the low resolution that we are looking at, and by the way, I forgot to mention this, but you usually use only the low angles when you perform the dummy atom modeling. So provided our limited resolution, we need to get a compact shape. So, and this is already some type of additional information that we can use during ab initio modeling to restrict the results to something that makes sense. Another type of restriction that we can use is symmetry. If we have prior knowledge about the type of symmetries that we have in our, let's say, protein, like in this case, then we can apply the symmetry as an additional restriction. And the process of fitting the data, so here in green we have the target function that we would like to fit, and the red is the scattering from the current shape. And as you can see, it very quickly approximated the target function, but it is still trying to find the most compact shape that has the P222 symmetry. So it's like cubic symmetry. Not cubic, but like, you know what P222 is, right? It's like P2 is like a dimer, and if you make a tetramer out of this P2, you will like get like the P22 symmetry, or P222, which is the same. So it's a tetramer. So in the end, so but a tetramer where like the monomers are not like a cross, but form more like cube, like this thing. So here you can see it better. So this is the Damin model from this tetramer with the P222 symmetry, and in blue and red it is the same tetramer from the X-ray crystallography data. So it's like two dimers are here in red and the other two dimers are in blue. So this actually gives you an idea how good the dummy atom model represents your protein of interest. So again, the model that comes in gray beads is computed from the small angle scattering data only, and the blue-green model comes from the X-ray crystallography data. It is possible to use Damin as some other programs on the grid. So we have a web interface where you can just log in, and very soon you will be able to log in there using the single sign-on that Alexander mentioned before. So you can log in there, upload your data, choose the symmetry that you want, and how many times you want the modeling process to be repeated. I explain you later why you want to repeat that process, and get the results. So the result will be sent to the grid, computed on the grid, and you will get the result by email. So the program Damin is a very popular program. However, there was a certain problem with it. It took ages to compute one model. So that's why we developed another version that implements the same idea, but has a slight differences in how it is implemented. So these differences permitted to speed up this program up to 40 times. So if you want to start your ab initio modeling, Damif is probably a better program to start. So a quick example, here we have a very small search space with just a few beads, and we have a current model in blue for Damin, and at each iteration step, Damin may change each bead, each dark blue bead to the solvent, and each light blue bead to the buffer. And the compactness of this model and interconnectedness will be applied and calculated as a separate penalty. However, in case of Damif, it will always have on each step a model that is interconnected and relatively compact. Besides, in Damin, we limit ourselves to a certain search space, which is typically a sphere of the size of D max. This has some side effects that normally you won't see, but for example, in this case, you see that the model touches the search space. So maybe the model would be more extended, but it cannot extend itself because the search space doesn't allow the model to do that. So with Damif, we don't have such a problem, and I will demonstrate you how Damif reconstructs the shape of a hollow cylinder. So it starts from a certain search space here in light gray, and it starts from a certain compact solid model here in light blue, and then the minimization procedure starts, and the search space is expanded if needed, and inside the search space there is always a compact interconnected model that will eventually be minimized. So the discrepancy between the given experimental data from a hollow cylinder and the theoretical scattering from the current model is minimized, and eventually you actually do get a hollow cylinder. Amazing, isn't it? I don't see that much enthusiasm. So Damif is also available as an online version. You can also download it as part of the ATSAS package, and you can run it both on the grid or on your computer. So this POS program's approach is called single-phase diameter modeling. What we could else do is we could make a multi-phase diameter modeling, which means that we could use beads of different colors. So here in this case we have a complex of 3 different bodies. So we could collect the data from each individual, like let's say it's a protein-protein complex. From each individual protein we could collect the data. We could collect the data of the complex where all 3 proteins are present. Besides, we could collect the data from subcomplexes, like where only 2 of the proteins are present. If we assume, and I must say this is a very bold assumption, if we assume that the structure of individual proteins does not change alone and in the complex, we could come up with an ab initio model which would show the complex, and individual parts of the complex would fit the data from the individual parts. So the program that implements that is called Monza. It's also a very time-consuming process. So to give you an estimate, for Damin, you need maybe a couple of hours to perform the modeling in the so-called slow or more detailed way. In Damif, you need mostly like half an hour tops on an average computer, and for Monza, you need like half a day, I guess. So, however, Monza permits you to perform ab initio modeling from things like protein complex with a nucleic acid, so protein DNA complex, for example, or it also permits you to work with neutron scattering data. And neutron scattering would be applicable if you do have a protein-protein complex or protein nucleic acid complex, but you do know that the shape or the conformation of the protein changes upon complex formation. So using neutron scattering, one could hide a part of the complex from the neutrons and make only the other part visible. So for proteins, there is yet another approach to modeling, to creating models of proteins. This approach is called dummy residue modeling. So proteins typically consist of folded polypeptide chains composed of amino acid residues. So the idea here is that we know how many residues we have. We can represent each amino acid as a dummy residue or as a sphere of a fixed radius. So, and in the program called Gasbore, one attempts to represent a protein as an ensemble of dummy residues. Each of these residues will be identical. They will not be connected in a chain, and they will be, what else? The number of these residues will be the same as the known number of residues that you know that there is in the protein. So this is another type of a priori information, like the number of residues in the protein, that can be used to create an ab initio model, which is, however, slightly more detailed than the dummy atom modeling approach. So the distances between these residues will be roughly the same, or the distribution in space of these residues will be roughly the same as the distances between the radius in the protein. And Gasbore will find coordinates of these dummy residues, again using some initial search volume. And you will get a higher resolution looking model. So typically when you perform this type of modeling, you also take higher angles into account, as long as they are not total noise. You can do that. And this way you get a slightly more detailed model. There is also a web interface for this program. So here you see what kind of information you need to provide. You can provide your input data, and you provide the number of residues that your protein has, and you can also restrict the symmetry of your model to a certain symmetry. So an important limitation of Gasbore is that it is applicable only to proteins, because otherwise the number of dummy residues wouldn't make much sense. So with the Damif you can also reconstruct the shapes of nucleic acids. So one of the problems with modeling of SAXS is validation of the models that you get. Let us have a quick look how we can validate our results in case of ab initio modeling. So before we start validating our ab initio models, we need to remember that we need to check the input data. So there is a simple rule with any SAXS modeling: garbage in, garbage out. If your incoming data is garbage, then your model will be just nonsense. We had a few years ago a very funny story at the beamline. So we were measuring some protein, and we have this automated pipeline that does all the data processing. And in the end, it also creates you an ab initio model. So that was one of the first implementations of that pipeline. Now it's a bit smarter, but back then it was just like taking the data, subtracting the buffer, and estimating all the parameters, calculating the P of R function, and then submitting it to the Damif program. So, and once there was a problem with the shutter. So the shutter wouldn't open. So the shutter wouldn't open, there will be no X-rays, but the detector would collect some noise some background noise. And then this noise would be subtracted from another noise, which was the measurement of the buffer, resulting in noise again. With this noise, it was actually possible to create a certain model that would fit that noise. Noise is very easy to fit, you know. And it actually resulted in an ab initio model. And the users were like looking at this model and saying like, oh, look, you can actually see this domain, that it is separated from that domain. And like the beamline scientist coming back from the hatch like, no, you cannot. You didn't even expose your sample to X-rays. So before doing any type of modeling, check for monodispersity, for radiation damage, for aggregation, for concentration effects. Check if the overall parameters, such as the radius of gyration, the molecular weight, the volume, they fit your expectations. Check that your data is not too noisy, and only after that you can proceed to actual modeling. However, if you even have decent data, remember I told you that you can come up with more than one shape that would fit your experimental data. And for certain shapes, the problem can be quite dramatic. For example, we took this flat disk, which has a P5 symmetry, and tried to reconstruct it ab initio, so like computed data from this disk, and tried to reconstruct it ab initio. And these are the models that we could get if we wouldn't put the symmetry restriction in. However, if we know that we are looking for something with P5 symmetry, putting the symmetry into Damif, it was possible to come up with much better reconstructions that would actually look very close to the original shape. So this is a more real-life example. This is an attempt of reconstructing an RNA using the program Damin. And here we have 6 models that all look quite different. However, they all produce an identical scattering pattern. So the way of dealing with this problem is to repeat the modeling many times. So if we repeat the modeling many times, we can superimpose all the models to see how different they are. So we can do that with a program called Subcomp, which superimposes models, normalizing the minimizing the so-called normalized spatial discrepancy, or NSD. So basically, it first superimposes or shifts 2 models together, and then tries to rotate one model against the other in a way that they overlap best. And then it is possible to compute this spatial discrepancy that would show how far one model is from another. So then we could fill a table that would show us. So we have n models here, and we can compare each model to each this way. And we could also calculate the average NSD value. And it would show us how different our models are. So this would give us kind of a measure of stability of the ab initio reconstruction. And I must say that this approach is also valid for higher resolution models. Besides, we could plot, like here, the spread region. So this is, if you superimpose all models, then you can find a certain search space, and inside this search space we could plot the density, how probable it is that the model is inside. In the past, it was also very popular to redo the modeling using this search space, so you know where your real model should be. Another approach, you can average all the models together and apply a certain threshold to this to this density to come up with a model that is the average of all models. However, this model will not necessarily fit the initial experimental data. So what is typically done is you run Damif 10 times, 12 times or so, and you run Subcomp that would compare all the models, and you find the model that is the most typical one. There is a model for which the average spatial discrepancy would be minimal. So the difference, like the most probable model. This model will fit the data, and most probably this model will be a good representation of your shape. Okay, however, it is possible that you would come up with, end up with 2 classes or then even more of different shapes, and this is actually not easy to distinguish. So, however, in this paper it was possible to find the final solution that was found within this spread region. And I think for an overview of ab initio modeling, this is enough. So if you have any questions, we have papers at our website that are available for download, and we have manuals for each of the programs. And besides, we have a forum where you can ask the questions and you will get the answer from one of the forum users. Questions either about the usage of the programs or about the data analysis in general. So, but since I'm here, I can answer your questions right away. So do you have any questions? Yes, please.
How do you treat flexibility in?
So, flexibility is an interesting topic. So in reality, nothing is really solid. So each protein is flexible to a certain degree. If the protein is slightly flexible, then you can still try to do the ab initio modeling. And one indication could be that the models would always come up in a very different way. You will get a very high NSD value. As a rule of thumb, the NSD value below 1 is good, NSD value above 1 is bad. And if you have a very flexible or intrinsically unfolded protein, then you cannot really model it, because this would mean that this protein exists in multiple conformations in solution, and you cannot really come up with one averaged model of it. So does that answer your question?
Yeah, so once we had another workshop, they gave examples that if you have, say, for example, a histag, which is just about 10 amino acids, and it's generally in structural biology, we use that to purify protein, but it's usually not very structured. And by taking that into account, you will be able to get a better fit. I mean, just treat it as a flexible part and then to model it. But during this process, we do not know a priori whether it's that tag has a more contribution or some parts of the protein, even say a helix is moving, and that affects this. So I'm just wondering, you also said we need to do a lot of runs in Damif, and then to use Subcomp to check convergence. So in terms of flexibility, is there a rule of thumb that up to until what extent we should just say this is too flexible to get a reliable fit?
So first of all, with a histag, if it is just like the histag is relatively short, and if you attach it to a megadalton protein, you can just ignore the histag. If you attach it to a 10 kilodalton protein, then maybe you should take it into account. However, typically something like a histag, you can just ignore. Besides, for ab initio modeling, it will just be not visible in the overall model. So if it is something as small as a He stack, you just ignore it. However, there are other methods to access the flexibility of your protein of interest. One way of accessing it is you can take a high-resolution model, do molecular dynamics on it, molecular dynamic simulations, and to see if adding this type of flexibility can improve the fit of your model to the experimental data.
One thing I can, already in the morning session, you mentioned using the Krekky plot, you can see whether they are flexible parts.
Now, if you see that from the Krekky plot, your protein doesn't have a peak, there is not much sense in making ab initio modeling.
Yeah, but if it's somewhere in between, you have a peak.
If it's somewhere in between, you can give it a try, and if you repeat the process 10 times and you get a high NSD value, then probably your reconstruction is very unstable. The reason for the instabilities could be you have a very anisotropic particle, or you have flexibility. So normally you know that's very anisotropic particle, for example, a very flat disk without prior information that it is a very flat disk is very hard to reconstruct ab initio. This is just a problem of the technique because it is using spherical harmonics, and the spherical harmonics are called spherical not for nothing. However, if you restrict your model, for example, you can restrict your search space to a very flat cylinder. Besides, all ab initio modeling programs have the so-called anisometry. And anisometry is either unknown, or it can be prolate or oblate. So prolate, if you set anisometry to prolate, it will force ab initio models to be elongated and would prefer this kind of models. And if you set it to oblate, it will prefer flat models. And this way it is possible to stabilize the solution. However, if the instability of the solution comes from the fact that the protein is unfolded, you should go a totally different way and forget about the ab initio modeling.
Yeah. Can you say a little bit about how you sample the coordinate space? Because even if it's a coarse-grained model, the number of independent variables are too many. Essentially, we could get caught in just without even getting to the minima. So how do you make sure that you get to a minimal point?
So in case of Damin and Damif, all the spheres, they're on a grid. So, and it's packed in a dense way. And the distance, or the sizes of the sphere, rather, you can vary as one of the parameters of the program. So you can use larger spheres, and you will kind of get a more lower resolution model, or you can use smaller spheres. However, even with large amount of spheres, if you do a brute search, or exhaustive search of all possible models inside the given search space, the number of these models is so high that if you take all computers that are available in the whole world now and start calculating, I think the universe ends, or the sun will stop shining before you are done with this search. So this is why we use the method called simulated annealing. So basically it starts with a model, shakes the model very, very much, and then tries to minimize it, and then shakes it again a lot and tries to minimize it, and so on. So, and this amount of how much it shakes the model, it decreases lower, lower, and lower. So this is called the temperature of the simulated annealing, this parameter. And there are a few parameters in Damin and Damif that can fine-tune this parameters. So like here you see that on the video. So you start with a certain initial model, and then it's shaken a lot, and then like it's minimized, but at each minimization step it's shaken again and again and again. So this is a solid way to avoid local minima. So simulated annealing doesn't fall into the local minima, this is the reason why we use it. Simulated annealing is also used for other type of modeling, like rigid body modeling, which I will show in my next talk. Does that answer your question? Yes, please?
You try to decide simulated annealing other optimization algorithms like standard evolutionary algorithms which are obviously related, but can be far more easily parallelized.
Like which ones, for example? Which algorithms, for example?
Just an evolutionary algorithm looks very similar to simulated annealing, but with a larger population, you can easily parallelize it.
Yeah. Well, not easily. So first of all, we do use the evolutionary algorithm in the method called EOM, the ensemble optimization method. There, the genetic algorithm is used. However, this is a bit of what I do. However, from the internal group discussions, I think simulated annealing is more suitable for finding the fit. So the genetic algorithm may not actually find the fit to the data, and I've seen cases where this happens. However, the simulated annealing always gives you a very good fit to the input data. With the parallelization, there were attempts to parallelize the program Gasbore. However, it could be demonstrated that the parallelization, due to probably the usage of simulated annealing, here I agree with you, is hard to parallelize. So it is possible to do it, but it gives like 10% faster results. So with the Damif, it was also shown that this doesn't make sense. However, optimization of just implementation of the algorithm and slightly the algorithm gave a much better result than parallelizing the old implementation. And however, parallelization is used in terms of you normally want to calculate 10 models in parallel, and this we use as a routine. So you never calculate just one model. You always need to calculate 10 models or 12 models. So, and you do that in parallel indeed. So this is probably why we wouldn't go further in terms of parallelization of the algorithm. I hope that answered your question. Any more in-depth questions? Good. Let us see how we can use this modeling on practice. So let's come back to our data. So let me plot it on a logarithmic scale. So this is data from the protein A. As you remember, we started the distance distribution analysis. So, and we created a decent P of R function. So actually, this is what is used as input data for the ab initio modeling programs. Not for all of them, but for Damin, Damif, and Gasbore, at least. So we do not approximate the experimental data, but we first do this regularization, making sure that it is actually possible to make a sensible P of R function, and then we approximate the this light blue data. So that's regularized data. So this is done for technical reasons. And the programs that do the ab initio modeling, they will read the file where we save this data, and they would read the Dmax, so this value, that would give an initial search space, and Damif will also read the radius of gyration to have an idea from which size of the model to start. So, and if we want to use the modeling, we log into the grid portal and we use the program, let's say Damin. Yay. And then we open the file where we saved our POFR function. It's called protein_a.out. We give it some project ID, and we say that we do not want any symmetry and we want the process to be repeated 10 times. So after that, you just submit this data. And in a couple of hours, usually, you get the result back as an email. So what happens here is this data is sent to 10 different computers somewhere in the grid where the Damin program is started, and then the results are collected back, and once all the results are ready, our server will check if the results are ready or not. It will collect the results, pack it in a zip file, and send it back to the email. We have a similar service that we run not on the grid but on a local cluster. So the grid is very good for high intensity computations. However, something like Damif can be actually started on a local computer, or we can start it on our local cluster. So as you can see, it's not that convenient because it has maintenance on the 20th of March, and the grid probably does not, hopefully. But anyway, let me just demonstrate to you how we do that here. So this is the online interface of the program called Damif, and we can choose the same file here and choose the units. We know that it is reverse nanometers. Besides, the program also gives us a hint that it's probably reverse nanometers. So we can choose one of the many symmetries that are available. However, we do not have any symmetry here. So we can choose the anisometry. So if we know that our particle is severely elongated or severely flat, we can use this one of these options. And we can either limit the search volume or use an unlimited one. So, and we can also choose how many times to repeat the process. Let's say we want 3 times. However, if we would choose 10 times, after all the 10 models are calculated in parallel, they would be compared to each other, and another algorithm would make clusters of typical solutions. So that is good to see. If you have 2 alternative solutions, you will see that you have 2 clusters, and each cluster contains very similar shapes, but typical shapes from each cluster would not be the same. So would not overlap very well. So this actually happens. Sometimes the shape of your protein is such that it is hard to reconstruct it back. However, one of these clusters would actually give you the actual model. So, and you should use additional information to choose which model is correct. So I will just put it 3 times to make it a bit faster. And if we stare at this page long enough, it eventually will show us that it is done with computation. So I pre-calculated this in order not to waste our time waiting. And this is what the result will look like. So it will show you how good the model fits the data. So here in red we have the theoretical scattering from the ab initio model, and in green we have the experimental data. So as you can see, it will always fit the experimental data. And these are the result files. So the result will contain obviously 3 models. Besides, it will also contain the fits. It will contain both the fits against the experimental data and the fits against the smooth regularized data. So, and all this data is available for download. So let us see what kind of shapes we got. So here we have 3 models for protein A and 3 models for protein B. However, I recommend you to run it 10 to 12 times in order to get a decent an NSD values that you can analyze. So this is our first model. That's the second model. And that's the third model. So, as you can see, these 2 models are very similar. However, this one is similar in terms of the overall size and kind of shape, but it's definitely different. So if you would repeat this process many times, you would probably getting more shapes that look like this or this, and just a few outliers that would look different. So let's discard this one. So I've actually done it, and I know that the resulting shape looks more or less like this. So, and now we can do something interesting. We can compare this to the actual high-resolution structure of this protein A that I happened to have. Oh my God, look at this. He actually managed to reconstruct the shape quite well. So again, this is high-resolution data from the X-ray crystallography, and this is the ab initio model that we constructed directly from the small angle scattering data from a sample that was randomly oriented in solution. Isn't that amazing? I don't see any enthusiasm here. Come on. So anyway, that again gives you an idea how good or how bad ab initio shape reconstruction is. So, and here you can see that actually it's not that bad. For a very low resolution method that is performed on a sample in solution, you can get the overall shape in just a few minutes. So let's have a look at protein B. So protein B is smaller and it's more globular. So you don't see that many differences among the shapes. So, however, if we compare it with a high resolution structure of protein B that I also happen to have, we can see that again it resembles the protein pretty well. So, do you have any questions at this point? Yes, please.
Actually, this looks more like the case we get. So you get this flat surface in a reconstructed shape. But in some papers, we saw that the reconstruction seemed to generate really amazing features, like the low resolution crystal structure. I'm just wondering, how did they do that?
We could try to do this with a program called Gasbore to see if we will get better data. So it will probably take a while to calculate a Gasbore model, but let's start it. So to get a Gasbore model, the first step would be to take the initial data and to repeat the POFR modeling. But this time we can take more like higher angles into account. Yeah, something like this. So this is here we take higher angles into account. There is some noise in there. However, we will pretend that there is not much noise. And if you take higher angles into account, then you can reconstruct smaller details, obviously. However, with Domino Damif, this doesn't make too much sense because it will look for first it will look for a very compact shape without too many features. And second, it will also reconstruct a particle that will be solid inside. Gasbore will not account for this homogeneity of the density inside the protein. So, and Gasbore will make a more detailed model. So let's save it. So that would be protein A high angles. So, and then we can start Gasbore. So we load our file with the data. We don't have any symmetry, but we need to know how many dummy residues to use. For that, we can open the sequence of the protein. So 1, 2, 3, 4, 5 multiplied by 80 plus 60. So it's 460 residues. So and we have various modes that we can use here. We can either fit the intensity or we can fit the POFR. Fitting the POFR is way faster. So Gasbore is a very slow program because it doesn't use the spherical harmonics but the Debye formula in order to calculate the scattering. So I will use this slower mode and hopefully we'll get a higher resolution structure. So I see here's also an option to fit the oligomer-monomer mixture. So there is an experimental version of the program Gasbore that would work on a mixture. If you can say that I have a monomer-dimer mixture, it will try to reconstruct a dimer such that in a mixture with the same monomer it would fit the experimental data. However, this is already a fishy approach. So let's see, maybe by the end of the day the model will be already there. That depends on how crowded the cluster is. So another possible answer to your question would be that something went wrong with the parameters and they just made a very fluffy model and they misinterpret this fluffiness as higher resolution. This is also possible. Yes, please.
I'm still not sure if I can get the right D maxima. I noticed that there's a quality estimate. There's a quality estimate when you check the distance distribution.
Yeah.
I'm not sure if I have to choose the D maxima with the highest quality estimate or not.
So, are you talking about this overall estimate parameters that we discussed in the previous talk, or
just this distance distribution?
Yeah.
And there's a quality estimate. See this?
Ah, this one.
Yeah.
Yeah. I think this is the same parameters that we got from the that we looked inside the file. It's a composite parameter, so it takes into account, I think, because this is already something that I have to look up myself in the manual. So, but I think it takes into account how good the smooth curve fits the experimental data, so the discrepancy. So if we try to just have an obviously wrong Dmax, we will have a worse fit here and a worse quality estimate. And I think it also takes into account some other parameters. For example, the PO function, it has Wobbliness parameter. So if you have a very, how do we do that? Okay, you quickly have seen an intermediate solution that was very wobbly, but anyway, if you have a very wobbly PO function with a lot of peaks, it will probably see that and take that into account in the quality estimate. Besides, here obviously we get a warning that the Dmax is too large for this angular range. Probably this is also taken into account. However, like with any other estimates, I would look at the overall data and to see if the data makes sense.
I mean, if you see the if you want to know if the D maxima is, for example, 9 or 10, will you use the quality estimate?
So, like, you mean if we have to choose between 9 nanometers and 10 nanometers, if the quality estimate for 10 nanometers is better, then probably 10 nanometers is the right answer. So this is roughly what happens if you click this autognome button. It will try various Dmax values for you and compare this overall, like all these solutions. And besides, it will also vary the alpha parameter that we do not have on the interface here. So the alpha parameter is kind of giving the algorithm a hint. Should the algorithm pay more attention to fitting the experimental data or to having a smooth, non-wobbly POFR function? So in a sense, if you press this automatic button, it will do that for you. However, this is not a strict quality estimate like other quality estimates. And in SAXS, unfortunately, there are no parameters that you could take and say like, okay, this solution is better than this solution because this parameter is better. You can say that if you have two models, one data set, and one model gives you a chi value that is smaller than the chi value from the other model. Then you can say, yes, this model has a smaller chi value, and it fits my data better than the other model. However, for example, this model may be disconnected, and it just doesn't make any sense. So you have to take these things into account. With the quality estimate here, I would take it into account, but I cannot give you rules that a quality estimate of 64.5 is good and the quality estimate of 50 is not good. So I'm sorry, but this is how things are. Yeah. Any more tricky questions? Yes, please.
You have shown the P5 symmetry disk and to fit with unsymmetry model and the P5 model. But what if I don't know the symmetry of the samples?
So usually you know the oligomeric state of the protein. And so for example, I have a paper where we had I think a heptamer, and it was a multi-domain protein that was not possible to obtain as a monomer. So what we could obtain was like a higher oligomer. The molecular mass estimates for that protein, again, it was not possible to distinguish between a hexamer, heptamer, and octamer. So what we did is we found a paper where the same sample was, or a very similar protein was investigated using the transmission electron microscopy, and there they were quite sure that it is a heptamer, so they could see it there. So that's how you obtain such information. In some cases it is not clear, and obviously you cannot guess it from the SAXS data, but sometimes such information is available. Yep. More questions on complementary methods? Good. Then I suggest we make a short break, and we will proceed to the final part of our SAXS tutorial.